Hi, everyone, and welcome to Scicomm Monday. If you're new to the broadcast, we try to make this as engaging a platform as possible. So please feel free to use the Periscope chat to send any questions and comments you have, and we'll do our best to answer them. I am your host, Nicole Wood. You can reach me on Twitter at WildlifeBiogal, or you can also reach me at the broadcast handle of Scicomm Monday. So today we have a special guest, Ramesh Laungani. He's a professor at Doane University down in Nebraska, which I admit before we started talking, I had never heard of it before, so I'm excited to hear about all the amazing stuff they're doing down there. So Ramesh, welcome to the show.
Thank you. It's awesome to be part of the program, so I'm psyched to be here.
So if you could, just tell us a little bit about your background and then tell us also about the university that you're now at so we can learn a little bit more about this wonderful little institution that you have down there.
Sure. So I'll do it in reverse order. So Doane University is a small liberal arts college in Crete, Nebraska. So that's about 30 minutes outside of Lincoln, Nebraska. And there are about 1,100 undergraduates here. We have graduate programs, but really those are more targeted towards like adult learners. So I work on the undergraduate campus. So everybody I teach is straight undergraduates. So we really are sort of a PUI. The way I got here was that I did my PhD at the University of Nebraska-Lincoln, so just 30 minutes up the road, in plant ecosystem ecology. And so I studied carbon and nitrogen cycling in grasslands. And so in graduate school, I knew going into graduate school, I wanted to end up teaching at a small liberal arts institution. And so as I was finishing up, the job market and the jobs that I was applying for were all targeted towards this type of position. So now that I'm here, even though I'm trained as a plant ecologist and studies carbon and nitrogen cycling, I've taught a variety of different classes, one of which being comparative vertebrate anatomy. I also teach conservation biology. That's a little bit more in my wheelhouse, but comparative anatomy, is a step outside. And a lot of times when you're in a small liberal arts college, you have to step well outside your own field and own comfort zone because that's just what the department needs. So here at Doane, there are 8 of us in the department. And so to teach the variety of classes we like, we have ecology classes. We don't have we don't have like advanced community ecology where maybe, at a bigger institution, you have, 8 ecologists on staff. Here we have 2. So we also cover a lot of the evolutionary stuff. So comparative anatomy has been super interesting to teach because I've had to become a student myself. I've had to start where my students are. But the plant ecology stuff, like I said, I study carbon and nitrogen cycling with my students. All the research that I do is basically driven by undergraduates and is all done by undergraduates as part of their curriculum in their biology major here. So we require all of our students to carry out experimental senior research. So my students walk in not knowing much about plants. Maybe they want to do something that's environmental. And the nice thing about a lot of the work that I do is it's related to topics like climate change. So students can hook into that topic really well, and they feel like they're really fighting or they're really dealing with a contemporary problem.
Yeah, that's one thing I heard you talking about is how much you're almost a student yourself because you're having to teach so many different classes that maybe you wouldn't if you're at a bigger university. Is that really one of the good appeals of being at a smaller institution like that, is you get to broaden your knowledge base versus what you would if you were really honed in on that one topic area if you're at that larger institution?
Absolutely. So I'll admit when I applied for the job or when I got the job, I was petrified because what is a plant ecosystem ecologist going to do teaching vertebrate anatomy? And when I found myself, I started paging through a textbook. I did some Google searches. I was like, what are the textbooks that are used? I started paging through textbooks. And as I was reading through the textbooks, I found myself, certain studies were highlighted here and there. And so I would look up those studies just out of sheer interest. And what I realized was I was learning more from reading those papers and sort of going off of using the textbook as a springboard. I was learning more about comparative vertebrate anatomy by using the textbook as a springboard than I was from the actual textbook. So I said, well, if I'm learning this way, if I'm learning all this cool biology this way, why don't I just do that with my students? So it's super great because I always feel like I'm learning that, and telling that to my students. What's also great about it is it breaks down the barrier of, oh, he has his PhD and I'm just, a lowly undergraduate. I can say, look, I'm a plant ecologist who's learning this stuff at the same, I just have a little bit of a head start on them, but there are things that I don't know about comparative vertebrate anatomy. I've had students explain papers to me. Like, that's been really great. We read papers in the class, and I'll be honest, sometimes I read them and there's a figure or two that I don't quite understand. So
yeah, I've
if the
been
student
there.
has a question, I have to be like, well, what do you think the figure means? So I have to fake it until I make it. And
yeah, that
what's
one
been
of
great
those
is
like,
that
please explain it to me, so like, because I don't know.
right, exactly. So like there was a paper we were reading about this super cool study where they gave monkeys trichromatic color vision by injecting an opsin gene, a human opsin gene, into their eyeball using a virus. Like, it's super cool. Yeah, like so these spider monkeys are dichromatic. We have trichromatic vision, and so the researchers were trying to understand basically when did trichromatic vision evolve. And so they injected this opsin gene into the monkey's eyeball, and within I think 20 weeks, it was able to see trichromatic colors, which is pretty crazy in a pretty short time span. But there were definitely some graphs where I was like, okay, I know these are important. I can tell they're important because they're figures, right? So if they're going to be in a figure, it's going to be at least relatively important. But I didn't quite get it. And then I had my students, as they were presenting the paper in class, they were like, and you see this part of the graph is where it can't see color. I was like, oh my God, that totally makes sense now. So it was a very proud moment. I try to aim for that, those moments where my students are no longer my students, where they're really teaching me, so we can switch roles. That's how I know I'm doing my job. So, yeah.
Yeah, that's one of the things I like about doing this broadcast. It's each week I'm being exposed to research that I never would have been otherwise, and so it really helps me to continue learning. And that's the fun thing about it, is there's so much cool science out there, and what we do, each individual scientist is such a small segment. It's very easy to kind of get trapped in your own science and not be exposed to all the wonderful science that's going on out there.
Right. And it's really easy to I was listening today to actually a podcast about imposter syndrome. And so when you start thinking about, when you're so hyper focused on your niche of research, it's really easy to feel like, oh, well, I'm not smart enough, or I'm not doing this the right way. Whereas, really you're doing great work. And other people need to hear about it. Like, you work on mute swans, I work on carbon and nitrogen cycling. We can learn stuff from each other. And that doesn't make either of us more or less intelligent than the other person. We just know a lot about a very small segment of the world. And so I think podcasts like this are really helpful in terms of hopefully even building some collaborative bridges to say, all right, how do mute swans impact carbon and nitrogen cycling? That's a different angle on the same ecological phenomenon.
Exactly. It helps you think outside the box, which is so nice.
Yeah.
And it brings new insights to your research that you might not have thought of otherwise.
Absolutely.
So speaking of your research, why don't you kind of dive a little bit more into it? Because
sure.
When we were talking about it before the broadcast, there's terms I've never heard of before, like biochar.
Sure.
Why don't you explain to everyone exactly what your research is and what brought it about? So.
Yeah, no, that sounds good. So like I said, in graduate school, I studied prairie ecosystems, and I was really focused on the idea of the impacts of species identity and how that affects carbon and nitrogen cycling in an ecosystem. So what I mean by that is, if you look at a grassland, it may look like a pile of grass, but there's a whole lot of diversity in there. There are a whole lot of different plant species. And you can imagine all of those plant species require resources from the soil, things like nitrogen. They're sucking up carbon through photosynthesis. But the question becomes, how different are these different species? So, for example, is grass species A really all that different from grass species B? And if they are really different, you can imagine if you have a prairie that's dominated by grass species A, it's going to change how those nutrients cycle. Or if you have a prairie that's dominated by grass species B, it's going to change how those nutrients cycle. But if species identity doesn't really matter, if nutrient cycling is sort of an inherent property of a system, then that's an important question to answer, especially when you're thinking about, well, restoration ecology. What ecosystem functions do we want to put back into this ecosystem as, let's say, we're trying to restore prairie? If the species identity matters, then we have to be really deliberate about which species we choose. If it doesn't matter, we could just chuck a whole bunch of species in there and see what happens. So what this led me to was at this undergraduate institution at Doane, nutrient cycling, unfortunately, is not the most interesting topic to an undergraduate. It doesn't hook them immediately. So I wanted to find a system that I could work with and that undergraduates could sort of sink their teeth into. So climate change, like I said, is a way to do that. And the way I tackle that is through this substance called biochar. So biochar is plant material. And if you think about plant material, it's carbon. But it's plant material that is cooked and basically burned at super high temperatures. So I make biochar at 400 degrees centigrade, but I've read papers where they make it in temperatures that are 1100 degrees centigrade. And you cook it in really low oxygen or no oxygen conditions. And you can imagine that making this biochar at various temperatures changes the chemical properties. So I put basically dead grass leaves. I even use used coffee grounds from Starbucks. I put that in a can. I burn it at really high temperatures, and that changes the chemical properties of it. And the way that sort of deals and fights climate change is because if you think about just a regular dead piece of leaf material, when it lands on the ground, it decomposes. All that carbon that's in that leaf material, most of it decomposes. So even though that carbon is stored in that leaf, in the short term, once it decomposes after, let's say, 5 or 6 years, all that carbon is re-released back to the atmosphere. So that plant material, while, yes, it sucked up carbon in the short term, isn't really storing carbon in the long term. As we all know, our activities are putting carbon dioxide into the atmosphere. And so we need to figure out ways to get that carbon out of the atmosphere. And so what making biochar does is it harnesses photosynthesis, but really sort of removes that decomposition process from the equation. So the plant takes in carbon dioxide through photosynthesis. We take that plant material, we burn it at really high temperatures. And now that carbon, when we chuck it on the soil, that biochar is really tough. It's thought to be inert. So that carbon that we put in the soil is now not going anywhere. That biochar is not decomposing. So after even 100 years, that same amount of carbon that you chucked in the soil is going to still be there. And so now what we're doing by adding biochar to soil, we are actually storing carbon. And that's how we are fighting climate change. We are sequestering carbon in the soil. So that's all fine and great. But the problem is what we don't quite have a grasp on is exactly what the impacts of biochar is on natural plant communities. So if we put all of this biochar everywhere and we're fighting climate change and we're super psyched about that, but this now encourages the growth of invasive species, or it causes the loss of diversity from a natural ecosystem, now that's an important trade-off. Now, that's not saying that we shouldn't use biochar to store carbon, but we're picking one function over another. And so what I do is I have my students, what's great about biochar, it's easy to make. The experiments are fairly simple in that we can take that biochar, we can mix it into some soil, and we can measure plant growth. We can measure how plants respond to that biochar. And so from an undergraduate perspective, the response variables are really tangible. They're really easily accessible. So the barrier to that science is really low, as opposed to something like nutrient cycling, where they're measuring how nitrogen moves around. That's an invisible process, so it's a little more inaccessible to a student. So measuring how plants respond to something like biochar is really nice. And what's really cool about biochar is there are a ton of different options. So I was telling you when we talked earlier about, you think of biochar like a frozen pizza. So if you take a frozen pizza and you put it in the oven and you bake it at 200 degrees for 10 minutes, that crust is going to have a certain set of chemical properties. If I take that same pizza and put it in the oven and cook it for 20 minutes at 400 degrees, the crust is going to be different. Or if I take a different pizza and cook it at those same temperatures, those crusts are all going to be different. So what's interesting about biochar is it's not all the same. So I use spent coffee grounds because, hey, we're going to toss it in the landfill anyway. Why don't we use this more productively? But what we don't know is biochar made out of coffee grounds going to behave and interact and impact a grassland ecosystem the same as biochar made out of dead grass or made out of corn? And so what's nice about all these small variations is students can hook into all of these small variations. They can vary temperature. They can vary what the biochar is made of. That's something called feedstock. So what do we put in the oven? They can measure. They can vary the amount of time that you cook the biochar. So again, the pizza oven analogy really works well, right? We've all burnt food, right? We've all burnt things in our lives.
I definitely have, for sure.
Right,
so.
Right. And there's a certain level of burntness that we can eat, and once you burn things past recognition, you got to toss it, right? So what's really great is it's sort of an endless combination of parameters and like conditions that you can make biochar in with the same set of easily accessible response variables. So the undergrads really hook into it, and then I can really I can get into this idea of carbon cycling and where is the carbon going and where is it being stored. So biochar is a really great way to get into the complexities of carbon sources and carbon sinks and decomposition and turnover rate. So I can touch on all of these complicated issues almost without the student realizing that they're doing it.
Great. Well, we had a lot of questions come in, so we'll try to get through them as best as possible. If we don't get to everyone's questions, please feel free to resubmit them during the broadcast, or definitely send them to our Twitter handles, because we'd like to be able to answer everyone's questions. So I'm going to try to get the best to get through all the questions that were coming through. So one of the first questions was, are you just working with native grasslands, or are you looking at any of the agricultural crops and how they could be impacted by things like biochar? I think is what the user was asking.
Sure. So there is a lot of work. Actually, most of the work in biochar has been done in agricultural systems. So I'm trying to understand how biochar impacts crop yield. So one of the other things that's been found is that when you throw biochar on the soil, it actually can increase the fertility of the soil. It changes the nitrogen availability. And as we all know, one of the things we do to make crops bigger is we throw nitrogen on the ground. So biochar almost acts like a natural fertilizer. So while in one context it stores carbon, in another context it can be used to help the fertility of an ecosystem. So a lot of the work that's out there already has been used in an agricultural system. It has been looked at in agricultural systems. What is missing is actually work in these natural ecosystems. So that's where that's the direction I took it in, because I'm thinking about it strictly from a carbon storage standpoint. If we have these grasslands that are sort of pristine, let's try to enhance their functionality by throwing this biochar on the soil. But a lot of the basis of my work, a lot of the things that have informed my work have come from agricultural systems.
Right. And that leads me to like the next question that came through. They were talking about other limiting factors, and I don't know 100% if the user was talking about if biochar can impact the other limiting factors when it comes to plant growth, but maybe you can talk about those other limiting factors. Sure.
So when we think generally about a grassland ecosystem, generally speaking, broad brushstroke, often a lot of grassland ecosystems are considered to be nitrogen limited. And so what that means is we have to throw, so you think about a cornfield, most of what's thrown on the ground as fertilizer is nitrogen, is ammonium. And so what we don't know is if we throw biochar on the ground in a grassland system, in a natural grassland system, maybe that enhances nitrogen availability. And so now phosphorus or some other element is limiting. Those are all questions I hope to answer. But yes, no, that's a really great question. This idea of multiple resources limiting plant growth, is a complicated one. There's a lot of great work. I'll just plug another experiment. So I'm involved in this experiment where we are adding nitrogen, phosphorus, and potassium to grassland plots. So this is not dealing with biochar. This is a separate set of experiments. It's part of a global collaboration called NUTNet, called the Nutrient Network. And what we do there is we basically ask, given that human activity is sort of fertilizing the globe, how do grassland ecosystems respond to this sort of eutrophication with nitrogen, phosphorus, and potassium? And how do communities shift and respond if multiple resources might be limiting? So what's cool about this project is the same methodology and the same types of fertilizer are being used at 100 sites all over the globe. And so you get these really powerful data sets. So I encourage anybody to look up NuTNet or Nutrient Network. There's some really great work asking fundamental ecological questions about how do grassland communities shift in response to changes in nutrient availability. So I'm trying to work on both fronts. The undergraduates there, they help collect some of that data through classwork, but they're not particularly involved in the research end of that, because like a lot of ecological studies, you throw fertilizer on the ground and it takes 3-4 years for a response to really manifest. And so unfortunately, that doesn't work out so well in the timeline of a senior who's trying to do a senior research project and graduate in a year. But what's cool is I've been involved in this project for the past 4 or 5 years, and you can actually now see. So what we do is we go out to a nature preserve that's about 10 minutes away, and I take my undergraduates out there and they help me fertilize the plots. They throw out nitrogen and phosphorus, and it's all factorial. So we have the different combinations of the elements. And what's really cool is now that we're in sort of year 5, you can actually see on Google Earth, you can see the outlines of the plot. So you can see these communities starting to shift in response to different nutrients. So all of my work sort of ties into this idea of how do plants respond to changes in nutrient availability. The biochar, like I said, is sort of the hyper accessible way for my undergraduates to get a taste of that.
Yeah, awesome. Like that's one of the things I always like, when you can actually have those big visuals to be able to showcase, like, this is truly how it's changing. Like when you're able to say, you can see it from Google Earth.
Right.
It really does like kind of hit home with people about that.
Yeah, and that was a surprise. I just happened to be on Google Earth, and I was like, let me see. I just Google Earth over my plots, and I was like, oh my God, I can literally see squares in the grassland. So it really hammers home the idea that humans can impact ecosystems in a variety of ways.
Yeah, I know. I was personally excited when I could see my mute swans and their nest from Google Earth. I was just like,
yeah. Absolutely.
Yeah. And it like really was able to like to showcase like the wetlands and stuff. So like things like that, it really does hit home. And it's stuff that I make sure I always include in my presentations when I'm going to a conference, or if I'm talking to the public. It really does help hit home.
Yeah.
So another question that came in was microbial functions of the soil. So does biochar impact that at all? Like, have you looked at that?
So the short answer is yes. The short answer, yes, it impacts it. The short answer is no, I haven't looked at it. And part of that is because those microbial, so in graduate school, when I studied nitrogen cycling, microbes, although I was looking at nitrogen cycling, which is fundamentally a microbial process, I kept microbes sort of in a big black box. The microbes sort of spit out nitrogen. I was basically in graduate school measuring how fast were microbes spitting out nitrogen and making it available to the plants, how much were they taking up for their own usage. So I wasn't really looking at fingerprints of what microbes were there or how that shifted over time. I was sort of looking at just rates of almost in and out. So absolutely. So biochar, when you add it to soil, so for example, like I have my students using coffee biochar. So coffee, if you mix it into soil, it's very acidic, right? So coffee is very acidic just normally. So when you're mixing coffee grounds into the soil, it's going to acidify your soil. So you can imagine that coffee biochar is also going to change pH parameters, which is also going to impact the microbial community. So I haven't had to be honest, I haven't had a student walk in because, again, my research is driven by my undergraduates, which is really the best part. But I haven't had a student walk in and say, I really want to study the tiny things that I can't see. I mean, the minute they do that, I'm on it. But I haven't had a chance to do that because the students that are walking in, I think they really like the tangibility of the response variable of plants. The other nice thing about plants as a response variable is they're cheap. So for measuring nitrogen, we have to collect all these samples, and then we have to pay for that analysis. We have to pay for those samples to be analyzed. And sort of smaller institution, funding goes up and down. Getting research funds for those samples, can be hit or miss. And so plant variables, a bag of seeds costs $5, whereas one sample costs $4. So if I have 100 samples, well, do I have $400 lying around? Maybe, maybe not. I'd love to answer, like, sort of look deeper and zoom in on the microbial impacts of biochar. Absolutely.
Yeah, it's one of those things, like sometimes you get so focused on something you forget like all the different like impacts. Like I'm an ecologist, even sometimes I still forget like all the different cascading impacts through all the different systems. Even when you're like in one spot, there's still so many different things that you can be looking at.
Yeah, and one nice thing about biochar is because it's hyper accessible to students, it gives me an opportunity, or gives them an opportunity, to think about the microbes, at least, to say, look, there's this microbial process called nitrogen mineralization, where nitrogen, ammonium, and nitrate are produced in the soil. So we're not only trying to understand how the plant responds, but we're also somehow getting sort of a proxy measurement of the microbial community. So this biochar experiment, being so simple, allows them to think across biological scales, from the plant level to the sort of ecosystem level, the scale of climate change, to the scale of like what's going on with the microbes.
Yeah, and speaking of climate change, someone had a really interesting question, was, while you're sequestering so much of this carbon in that biochar, how much are you releasing by burning this plant matter?
Right. No, that's a huge thing. So generally, when you take, I'll just use some even numbers here. So if I have 100 grams of plant material, generally when you burn it, you lose about 50% of that plant material. And I haven't done the full accounting of how much carbon we're releasing when we actually flick on that oven. That's something I'm going to have my research students do this fall, actually. But let's just, I know this is unfair, but let's say the actual flicking on of the oven releases 0 carbon. I know that's not fair, but so when you burn the material, you lose about 50% of it. And while that seems not so great, the 50% that you keep is now there, as opposed to adding, let's say, 100 grams of just plant material to the soil. That plant material, in 4 or 5 years, or even 10 years, again, not a very long time on ecological timescales, most of that, 80 to 90% of that carbon, is now back in the atmosphere. So while you lose 50% of the carbon on the initial making of the biochar, the 50% that's still there is now there. It's not going anywhere, or very little of it is going places. And again, there's a lot of work on testing this idea of, is biochar really as inert as people claim? And again, does that change with temperature? Does that change with feedstock type? So again, there's sort of this endless amount of questions that can be answered looking at biochar. But yeah, that's definitely a concern as I came to this system. Are we actually having a net? Are we not actually storing any carbon because we burned off 50% of it and the oven has released more carbon than we're actually storing? But yeah, no, that's definitely a concern, and especially at large scales, that has to be taken into consideration as we think about like the whole carbon footprint of a study like this.
Yeah, because I think that's something that I kind of forget about is, plant materials or other things decompose, they are releasing that material, and it's not just necessarily going into the ground. Some of that is being released up into the atmosphere. And yeah, so everyone while everyone says plant a tree and it helps climate change, absolutely, right? But again, at the scale of geologic time, a tree that only holds onto its carbon for 100 years isn't really doing much, and I'm going to get these numbers wrong, but we hear, nowadays, like, yeah, the carbon that we're seeing in the atmosphere was first burned in 1950 or something like that, So a lot of carbon has really long residence times. So what we're trying to do is leverage the fact that you can have carbon remain in a pool, in this case, the soil pool, for really long periods of time by eliminating that decomposition process or by reducing it severely. And I think, again, that's a process, decomposition. I always tell this to my students.
think about what the world would be like if decomposition didn't happen, right? We would just have piles and piles and piles of, I spend hours raking my oak tree leaves every year at my house, and that's all plant carbon that is going to waste, right? And if that plant material didn't decompose, what we would be swimming in dead leaf material, right? Society would be very different if decomposition didn't happen.
Oh yeah, I mean, how would you get from place to place?
Right.
At least on like the very minimum, as it's like compressed down, you still would be like a good
Right.
mile or two up in the
Right.
air from all of this.
Absolutely, absolutely. So again, this biochar topic really helps students visualize and think about this again, this invisible process that people don't think about, the importance of decomposition. Um, although although I'm not a microbiologist by training, my colleague who's a microbiologist, she always says microbes make the world go round, and I fully agree with that. I'll say microbes in plants, but yeah, like those processes are fundamental to ecosystems and how they function.
So another question that came in is you're an undergraduate institution, but are you able to bring in postdocs or anyone else above? Like if you need that additional help, or is it just completely limited to, at least in your department with the undergrads with research?
No, we can. So, but that is, but we have to have funding for that, right? So we have to have the time to write the grant for the postdoc. So those opportunities are there. One of the challenges at a PUI is that, especially one where research is like encouraged, so at my institution, research is strongly encouraged, as it's really thought of as a pedagogical tool. So for lack of a better phrase, I don't really care what my students find, because what I want them to do when they're in the lab, I want them to figure out how to screw up in the lab and how to think critically and how to organize a data sheet and how do you have to really think on your feet. So I think of all these experiments as teaching them how to do science more than worried about, am I really going to solve this biochar problem? And so because of that, teaching is really the focus. And so if I were to write a giant grant and get like a couple of postdocs that were, really focused on these research questions, unfortunately, that would pull me out of the classroom. And that's not what I want to do. I don't want to get a course release that now makes me answer a question about nitrogen cycling, and now I can't teach my students. So the great thing about these research projects, because they're hyper accessible, it becomes an avenue into science in general. And again, the barrier is the response variable barrier is super low. So students can get data. They feel like they've accomplished something. Like just the other day, just Saturday, my students came in. We spent like an 11-hour workday harvesting plants. I think I sent you a couple pictures of that, harvesting plants. And, we were clipping these plants. I said, the minute you clip those leaves, we have data. You guys have done science. And these were freshmen, so they just finished their freshman year. And I think it's those sorts of experiences that are really critical for keeping students in the pipeline, right? So that science has to be something that they can do. It's not something that, I have to get my PhD. Oh, the professor's really smart. No, can you clip some leaves and dry them and weigh them? Great, you can be a scientist, right? So yeah.
Yeah, that's a big thing that I really wish more institutions would do, is make sure that their undergrads get that experience, because when we were hiring people in for the projects that my lab runs, it was always such a great benefit is if we could have undergrads who actually already had research experience, and we knew that, okay, you're much more likely to be able to handle this, rather than having someone fresh off the boat kind of,
Right.
all right, we've got to completely train you from scratch, which, I mean,
Yep.
can be nice with some instances, but a lot of times it's nice to know that they've already got a little bit of that experience and you don't have to completely go through the whole process of training them.
Right. And, what's really important is that they have that grit to say, yeah, things are going to go wrong. Things are not going to go, or going to go wrong, or not going to go as expected. And a student needs to have the experience to be able to say, yeah, this is just part of the process. Whereas if they're getting that first experience in a high pressure lab, that's maybe where they say, you know what, I can't do science. And that's where they leave science, when in fact their screw up really is just part of the process, but they've never been exposed to that idea, right? because they think of science as this end product, like I have to determine a fact, or I have to cure a disease, or I have to like fix climate change, or and so having these experiences really help build in pathways for undergraduates, I think, and build in that grit.
Yeah, no, that's a great point, because I know with my very first trip out for doing my mute swan research, when we were actually collecting the plants, I ended up having to throw all the plants from that first initial trip away because of the way we were sampling them. I didn't realize it until like as we were out there, I was like, this is not a really good quality sampling method. We need to like redraw this. And my parents, who were amazing that year and were my actual field techs, I mean, I completely lucked out. My dad just looked at me, he's like, wait, all that work is getting thrown away? What are you talking about? But it was like, I mean, that's the process of science sometimes, is you are going to make, those mistakes, or just, realize that, hey, maybe there's a better way to do this. Let's improve upon it. And then we'll go with that. And
absolutely. Yeah. There's there was a great analogy I heard that science, is presented as this very linear method, linear process, right? Hypothesis, the scientific method we all learn, but it's really fumbling around in the dark. And so that's really what it is, fumbling around in the dark in an organized manner. I also compare it to trying to look into a cave with a pen light, so you can't see, you don't even know it's a cave. So you can just look at small portions at a time. You collect all this data and you say, yep, this looks like rocks. And okay, maybe there's some bear poop on the ground. So yeah, maybe my hypothesis is this is a cave. Here's the data, but you can never see the full cave. You just have to piece all those pieces of data together to draw that conclusion. So yeah.
Yeah, especially too, like sometimes you don't know until you're actually out in the field or running that experiment what's going to work and not. Just because it worked for someone else, it could be an exact replication of somebody else's work, but maybe the system that you're in, it's just not going to quite translate over, and you just don't know until you're out there. You can do all the homework you want, and it's like, nope, this isn't going to work.
Well, and we all kind of know that method sections of papers are just suggestions. We know there's just a ton of details in there that's like, oh yeah, so we sampled the soil. Well, how did you sample it? Oh, well, we had to get these special tubes. We didn't put that in the detail, So yeah, we all have read papers where we know the methods are just like, in a perfect world, where you work in my lab, we'll get this.
Yeah, exactly. So one of the other really cool things that you are doing that I'm really interested in is this 1,000 STEM women project that you're working on. So can you tell us about that a little bit more?
Yeah, sure. So this past year, I've been interacting with a 6th grade classroom in Rhode Island, and I use this really awesome tool called Flipgrid that I suggest, if you haven't heard of it, I suggest Google it and look it up on Twitter. It's amazing. But basically, what I would do is I would post every week. I would post a short video prompt to these 6th graders about some ecological question. So, for example, one week I asked them, how many different plants and animal species do you need to keep an ecosystem healthy? And I deliberately made them sort of open-ended. And I got these and Flipgrid allows the students to respond with short videos. And, they're 6th graders, so they're like absolutely adorable responses. And so it was great. I was having this conversation with 6th graders in Rhode Island, this sort of asynchronous conversation with 6th graders in Rhode Island about science. And so the last prompt that I made was I asked them, okay, they've learned a lot about ecology. So I asked them about, diversity. I asked them about pollinators, all these things. And I said, what other types of scientists would you want to interact with or meet? And, a lot of them said marine biologist and astrophysicist and chemist. And while that's great, what I recognized was that they were naming scientist types. They were naming large groups of scientist types, and they were naming scientist types that you'd see on the Discovery Channel, or like every day on the news, right? And again, while that's great,
yeah, I mean, I can say I did the exact same thing when I was in 6th grade. It was like what I saw on the Discovery Channel. I wanted to be either like Jane Goodall studying apes, or I wanted to be the marine biologist going out and studying orcas. That was just what you do when you're 6th grade.
Yeah, exactly. And so I started thinking about this idea that science is so much more diverse than that, right? And when graduate students or professors talk about the science they do, they're not saying, they say, sure, I'm a marine biologist, but then they immediately zoom in to talk about the part of marine biology that they work on. And that gets, I think, lost. That sort of specialization gets lost to those students. And what the problem with losing that specialization is it doesn't provide the students with a broad picture of all the ways you can be a marine biologist or all the ways you can be an astrophysicist. They probably think of like, oh, well, if I'm a marine biologist, all I can study is Shark Week. All I can study is sharks. But so what I wanted to do with this project is Flipgrid makes these short 90-second videos, or allows people to make these short 90-second videos introducing themselves. And so what I want to do is sort of curate this library of 1,000 female scientists that they introduce themselves and they say, hey, my name is, and this is what I work on. And I also want them to talk about what's left to figure out. What don't we know? And then these short 90-second videos can be this archive that any K-12 classroom can access to now say, hey, go watch 10 of the videos on, 1,000 STEM women and write a reflection about which one was most interesting and why. So a good friend of mine, Dr. Rachel Burks, she's fairly well known in the Scicomm world, but she in her video, she highlighted the fact that she's not just a chemist, but she's an analytical chemist. And again, it's that specialization, and she highlighted what an analytical chemist does versus what a physical chemist does. And again,
she even went into the fact that she does like forensic, chemistry. It was so detailed, like kept going like down like a whole nother like step in like the ladder of like what it is that you do, and it was really cool to hear.
Yeah, and so for a sixth grader hearing that, they can now say, oh, I didn't realize that was a thing. That's really the goal. I want the sixth grader to say, I didn't realize that was a thing. And while I want the scientists that are involved in the project to target, like in their language, sixth and seventh graders, this is a resource that could be used at university classrooms, sort of all over the place, right? Because how many college students walk in and say, I like science, but I don't know what I want to do, so I guess I'm going to be interested in pre-med, or I want to, and this isn't trying to dissuade anyone from becoming a doctor, but, they don't know what the options are. So my hope is to curate this library that's now there. It's this archive that is just there, and now anybody can access it through this link. So this is my shameless plug for trying to get female scientists to make their 90-second video. It's also, I guess, a shameless plug for Flipgrid. I don't get paid by them. Let me be clear. But it's like, for lack of a better, I use it in all my classes. I call it my drug of choice at this stage. It's really, really great. It's really helpful in all my classes. But it's really great, I think, in this context as well, to sort of build this library of experiences that can be shared so easily over the web.
No, I think it's a really good project, because I think of, my niece who is 7 years old, and if she didn't have her scientist of an aunt,
right,
what would her ideals be of like what different careers that she might want to have growing up? And she recently wrote on one of her homework projects, this last school year, oh, I want to be an ornithologist.
Right.
What, first graders going to say they want to be an ornithologist unless they've been like directly exposed to it, because most first graders probably wouldn't be exposed to either, they might go out birding maybe, but, even if that, they're not going to necessarily know what an ornithologist is. But here she is, writing that down, and you're helping get kids exposure to things that they might not otherwise have. And it's super important to have that.
Right. And, I've run a number of times this little experiment of like the draw the scientist experiment. What do you get, right? You get bald white male crazy haired white male lab coat, right? And obviously we all know science is so much more diverse than that. And so this project is really motivated to try to get at and try to break down those stereotypes, while also showing students that there are all these opportunities out there. There are all these ways that students can fit in to science that aren't simply what you see on the Discovery Channel. And again, they don't have to understand every detail of what an analytical or forensic chemist does, but they have to know that, yes, chemist is a broad umbrella that a lot of different stuff goes into, or ecologist, or marine biologist, or, there's a lot in there. And so my hope is and I know for me, when I saw an Indian ecologist, that helped me visualize a path like, oh, I can do this. I'm originally from New York, so the idea of, when I was 18, the idea of me living in Nebraska at the age of 36 was sort of, what are you talking about? But so my hope with this sort of Thousand STEM Women library is to, humanize a career path, right? So that a student can, and there's plenty of research, right, showing that if students can see themselves in somebody, they can see themselves in a career path, they're more likely to go into it. So my hope with this is, I started with 1,000 STEM women, and if we can get to 1,000, then maybe we target other demographics, right? But I thought, well, I've never done anything like this before, so let me start with at least 50% of the population and see what I can get. But as you know, it's fairly easy to make a video. So whoever is watching or listening, I strongly encourage you to do that. Make a video, go to Flipgrid. You can even do it on your phone. You have to download a free app if you're going to do it on your phone, or you could do it on a laptop without an app. So I have a bunch of details. There's a web link that I sent to you that I'm sure we can tweet out. That goes into the details of how to do this, how to get involved in the project.
Yeah, like I'm showing everyone right now the video that I just did this afternoon and uploaded, and it was so easy and fun. Like, it probably won't take most people as long as it did me, just because I'm a perfectionist when it comes to, science communication, but it's so easy. You can either like upload it directly, by making your own, or you can just record right on the website there. So like, you don't necessarily have all the like fancy software that I do to be able to do these things. You can easily do it on your laptop, and it was so simple. And you have all those examples up there already. So like, I watched a couple of them to see kind of like what it was that they were doing, and it was super easy, and it was just have fun.
Yeah, and the way I also think about it is this is professional development for us as Scicommers, right? So if I'm a professor or a graduate student and I want to learn how to talk to a seventh grader about my science, I work on the rule that most people understand most topics at a seventh grade level. And that's not saying they're dumb. That's just saying that we all don't have time to have PhDs in everything. So, if someone wants to get involved in the project, they can really also think of this as not only helping future scientists, but helping themselves in terms of how do I translate my science in a way that a sixth grader can understand, that isn't jargon laden, that isn't, if I study cell respiration, do my students, do the sixth graders really need to know that I study phosphofructokinase, or do I need to explain that I try to study how the molecule that we use for energy, glucose, that we get our energy out of, changes shape, and I study one little step in that process. So this project is also meant to help scientists practice their own science communication skills.
No, it's definitely a key thing, because I know like there was a couple times, like as I was doing some test runs with it, like it's hard even for me, who communicates a lot with people, to not just randomly start throwing in some jargon and making sure like I tone it back, because I like to, whenever I talk about my science, I usually try to filter it through my niece and nephew. Whenever they spit back at me is how I put it out, because I know that, say if a physicist or even another ecologist that might be studying something slightly outside my realm, like I need them, like, quote unquote, to like dumb it down to my level, because I just don't know it. And that's, I'm a scientist and I still don't get some of the science out there.
Absolutely. So. Right. Like my understanding of the Higgs boson is pretty much at the level of, the Big Bang Theory in seventh grade,
Exactly. So, if it wasn't for the Big Bang Theory, I probably would not even come close to even knowing what it was, much less how to understand it. So these opportunities, I think, need to be more opportunities like this. I'm not saying just the 1,000 STEM women, but as scientists, we need to become better at communicating not only what we found, but why is it important that we study the impact of biochar on grassland ecosystems, right? You could make that sound really dumb and really, tiny, or you could say, this is an important question if we are going to fight climate change. Those science communication skills take practice, right? And, I always think, how do you get to Carnegie Hall, right? Practice, practice, practice. And so, I feel like a lot of the younger generation of scientists believe in science communication. They understand the importance. They see the growing divide between sort of scientists and the public around a variety of topics, whether it be climate change or vaccines or any of those things. And so there's this growing divide. And so we got to start building those bridges. Scientists can't just, stay at conferences telling each other that humans are causing climate change. We need to get out there and show people how we reach those conclusions, and explain them in a way that doesn't necessarily need you to be a climatologist, that doesn't necessarily need you to understand climate models, right? That you can understand, hey, can you go out and see when things are flowering? Is that changing?
And we need to work on those skills as a scientific community. And we need to foster those skills in our graduate students, in our undergraduate students. And so projects like the 1,000 STEM Women are really trying to get at a lot of that as well.
Yeah, no, I completely agree with you, because I'd like to think of, if I was to actually go back and watch some of my very early Scicomm Monday episodes, or maybe even back before I started Scicomm Monday when I was doing some initial periscopes on my own personal channel, like if I go watch them, I might just kind of twitch at the awfulness of what they are compared to where I am now. But it is like, you just have to practice and keep doing it to be able to get better at it. And eventually, I've been doing Scicomm Monday for about a half a year now.
Right.
And just that practice alone, like I've been able to see some improvement. I'm sure if I really truly went back, I'd see a lot more improvement than like what I want to admit has happened. But you really you need to just keep going and doing it. And if it's awkward, that's great. You eventually will get through it. Like I know one of my old habits is I like to say, you guys, instead of like, oh, this is, what your project is. And it's just like little verbal foils that I have that like irritate me, that, it's taken a lot of practice to try to get them out. And
Absolutely. Yeah. Yeah.
But all right, so I'm going to throw up the link here for everyone so they could see it. It's www.doan.edu/1000 STEM women project, and I highly encourage all the women in STEM to go out there and make the videos, because it was really simple, it was fun, it was a great excuse to be able to just talk about your work. And what's nice is knowing that like there's going to be some kid out there that's going to see that and maybe be able to go, hey, that sounds really cool, I want to do that. And otherwise they never would have been exposed to it, otherwise. So
right, absolutely. And what better of an ego stroke than can it be than to have a sixth grader say, oh my God, I want to be you, 10 years from now or 15 years, right? Papers are great, right? We're all happy when we publish a paper, right? It's a great ego stroke, but we all know that it's going to get swallowed, whatever the stats are, right? 3 million papers come out every year, or whatever the stat is, right? But if there's a sixth grader who sees a 90-second video and say, yep, I study crabs, or I study burnt coffee and how that affects grasslands, and now they go and solve this problem, selfishly speaking, right? Great. I can claim that academic pedigree, right?
I know with my niece and nephew, the fact that both of them have like in their career goals that they want to be a scientist somewhere along the way. My nephew still wants to be a pro baseball player, but he's bound and determined he's going to go get his undergrad first, in some sort of science, go have a 20-year MLB career, and then after that he's going to like go back to science and do research. The fact that like both of my niece and nephew think that I'm cool enough that they want to include that in their future goals, that's awesome. That is Absolutely. Like you said, a big, like, it just makes me feel good every day.
Right. And the other thing is there's so many students that don't get exposed to scientists on a daily basis, right? So the only scientists that they run into are probably the closest thing is a healthcare professional, right? Yeah. A healthcare professional or their science teacher that they can really tangibly connect with, right? Again, Discovery Channel is great, but there's that screen barrier, right? So now again, the 1,000 STEM Women doesn't solve that particular problem, but it at least tries to diversify and minimize the barrier there.
Right, and it's also just inspiring to me as well to like keep doing great science and keep doing awesome science communication, because that you're helping to inspire that next generation of scientists, or maybe even scientists within your own generation, to go out and keep doing what it is they're doing, and you just keep building everyone up, and it just makes a better community overall, I believe.
Yeah, and what I'll do is after we're done, I'll share a couple of the responses I've gotten from the sixth graders that are, again, like, just, they were so, I joked around with my colleagues, some of them were so adorable. We're like, we should start recruiting them to come to Doan right now.
Right. Hey, sports guys do that. What is it? Urban Meyer and, Nick Saban from Alabama, they're recruiting 6th and 7th graders now for their football teams. Why can't we do the same thing for science?
Absolutely. So I'll share a couple of those links with you after we're done. Yeah.
Yeah, that would be fantastic for sure. Well, I think we're just about out of time here. So, Ramesh, any last thoughts you want to share with everyone?
No, I guess I would say the more you can get students and undergraduates involved in science, do it. The more that they can take ownership of those questions, do it. Yeah, and please, please, please get involved with the Thousand STEM Women Project. It doesn't take that long. My hope, we're at 10, so we're 1% of the way there. So my hope is to get the ball rolling, and then my hope is to have things just roll in. Check out Flipgrid as an app on your phone. It's super helpful. And yeah, no, this was awesome. I'm so glad that we could connect doing this, and I'll let you know what happens with the coffee ground study. Like I said, my students just harvested that data Saturday, so it's in the drying oven right now. So I will. So we have to weigh the biomass and see whether these different plant species responded to different biochars differently. So I'll keep you posted on that.
Yeah, that'd be great. And yeah, like, anytime you've got new results coming in from your research, or if the Thousand STEM Women starts to blow up and you want to come back on and talk about it some more,
Absolutely.
I mean, we're definitely game for that. So
Yeah,
this isn't
absolutely.
just a one-time only, stop on
Yeah,
Scicomm Monday.
that sounds great. That sounds great. Awesome. Awesome.
Great. Well, for everyone out there, if we didn't get to your questions, I know that there was probably a few of them that slipped through that I didn't quite answer. Definitely feel free to talk to Ramesh with his Twitter handle, which is at Dr. Ram Bio. So pretty easy to remember, nice and short for if you have any long questions for things. And then if you have any questions for myself or for the broadcast, you can reach me at Wildlife BioGal, which is my personal handle, or at the broadcast handle of Scicomm Monday. And I just want to also send a shout out to everyone for let's see, 2 weeks from now. See, it's already getting like where the summer is just like hard to keep track of where we're at. But 2 weeks from now, we're going to be at the American Ornithology Society conference because I'll be presenting there about science communication. So I'm going to bring Scicomm Monday along with us. So stay tuned for a bunch of different broadcasts that will be coming possibly fast and furious during that conference. As anyone who's followed me at different conferences before, sometimes that you'll get maybe only 5 minutes heads up that there'll be a broadcast coming on, but we're going to try to showcase a bunch of different researchers. We'll also be broadcasting not only one-on-one interviews with them, but also some of their presentations that they're going to be giving, whether it's their posters or their talks. So stay tuned for that. We will be off next week. My niece and nephew, who are awesome scientists, or beginning scientists, will be over here. So I'm going to be hanging out with them and taking them and helping them explore the wide world out there. So we'll be off next week, but definitely tune in for updates when it comes to American Ornithology 2017. And I also want to make sure I thank all of our Patreon sponsors. We really appreciate all the help that you're giving to help keep this broadcast going. It takes a lot of work to put on the weekly broadcast, from time to equipment to just everything in between. Any help that you can give would be most appreciated. So please go to our Patreon site to help assist us with keeping this broadcast going, because we like sharing all the amazing science that there is out there, and your help goes a long way with making sure that continues to happen. And with that, I just want to be able to tell everyone thank you for tuning in today. Go out, do some science, have some fun, and we'll see you on the next Scicomm Monday.